electrophoresis. Gel electrophoresis is used to compare DNA fragments. The DNA is placed into the gel chamber, which conducts the current from a power source. This causes the DNA fragments of different sizes to separate and travel towards the other end of the chamber. Gel electrophoresis is used in areas of science like forensics and molecular biology to compare a suspect's DNA or things such as blood, proteins, and gunshot residue.
This is the equipment you need for gel electrophoresis. First, you need a solution to make the gel, and then you need a tray with 2 dams on the side, and inside the tray there's a ruler that measures how far the bands travel from the DNA. And then you need a comb that you put into the gel to make the wells so that the DNA can be inside the gel. And then you need a gel chamber, which you put water in from a beaker. And you need a power source to hook up to the chamber to run an electric current through the gel. And then you need a pipette to transfer the DNA into the wells in the gel.
Now I'll show you the procedure. First, a gel is made by mixing agarose powder and water. The mixture is heated and then poured into the tray. Once it cools, it forms a solid, just like Jell-O. While the gel is cooling, pour your water into the gel chamber to conduct a current. After the gel cools, remove the dams and the comb, then place the tray containing the gel into the gel chamber. Use a pipette to transfer the DNA samples into the wells made by the comb. Then connect the gel chamber to the power source and turn the power source on. The power source runs a current through the gel and the DNA and makes the DNA fragments move towards a positive electrode. The fragments separate based on size, so the smaller ones travel further. Once the fragments have reached the end of the gel, turn the power source off and compare your DNA samples.
Once the DNA bands have reached the end of the gel tray and the power source has been turned off, remove the tray from the chamber. A solid block of gel will be left with the DNA bands that have traveled different distances. The ruler on the side of the tray can help measure how far the bands have traveled. The DNA pattern of each sample can be compared in order to eliminate or identify a suspect. The next video will provide more information.
So we're going to go ahead and draw a gel, and I'll label everything for you. All right, if this is the negative end and the positive end, remember we're going to load our DNA at the negative end because DNA samples are negatively charged. Okay, so those represent the wells, and as the electrophoresis reaction is occurring, the negative current here is going to push the samples toward the positive end. And so you may end up with something that looks a little like this. Okay, and so each of these lines represents a band of DNA. All the pieces of DNA in this band are the same size. And so you can see here, everything is this distance, is the same size. Everything here, all the same size. These here, even though we have two different samples, are approximately the same size fragments. And so those fragments that are the smallest are going to move the farthest, and those fragments that are the largest are not going to travel as far. And there's a reason for that, and that deals with how agarose is made. Depending on the concentration you have of agarose, it's kind of like a mesh. Okay? I like to use the analogy that it's like the trees in the forest are really close together, or the trees in the forest are farther apart. And so if you have a really far apart forest, the bigger pieces will be able to move a little bit farther. So if you're on a horse in the forest, that horse is going to be able to move fairly easily through the forest. If you're in this forest, where the trees are much closer together and you're on that same horse, you're going to move a lot more slowly. And so if you have a dog with you while you're riding through the forest on a horse, here the dog's going to move even more quickly than the horse because it's smaller. And down here, the dog's going to move much more quickly. And so we have our large fragment here and our small fragment here at the positive end of the gel. And then up here where the gel is less concentrated, so for example, this would be a 0.8% gel, and this would be a 1% gel. That horse is going to move a little more farther down the band, or down the gel, and then the dog, the smaller fragments, are going to move very far. And so that's the difference you see between your two different concentrations of gel, and in general, how this works.
The case of Earl Washington made gel electrophoresis prevalent to the scientific community. Go.
In 1984, Earl Washington was convicted with the rape and murder of Rebecca Lynn Williams in her apartment in Virginia. He had an IQ score of 69, which is classified as mental retardation. 22-year-old Washington was arrested for alleged burglary and when questioned by the police, confessed to the murder of Rebecca Lynn Williams. Washington knew little about the victim. He was not knowledgeable about her race, height, or where she was killed. He did not even know she was raped. A rare plasma protein was found from evidence at the crime scene, one that Washington did not have. But the forensics report which tested this protein was deemed inconclusive.
On January 20th, 1984, Washington was sentenced to his death penalty. DNA test results were discovered in 1993, showing that Washington didn't match the evidence at the crime scene. However, the governor at the time only reduced Washington's sentence to life imprisonment. Washington's counsel continued to push for another look at the evidence and eventually convinced Governor Gilmore to allow additional DNA testing. Washington tested negative in comparison to the DNA sample found at the crime scene. After 17 years on death row, Earl Washington was pardoned of his capital murder conviction in October of 2000. After being exonerated, Washington was given almost $2 million for his wrongful convictions and imprisonment. Washington's case was the cornerstone of a new Virginia law which allows inmates to seek DNA testing if they believe they have been wrongfully convicted.
The next video will provide more information.
Did you kill that woman?
No, sir.
But you told the police that you did.
Yes, sir.
Why did you tell the police that you did it?
I don't know.
You don't know.
No, sir.
Did you understand then that you were being accused of a murder?
No, sir.
You didn't understand that.
No, sir.
When the police officer was asking you questions, what did you think?
Oh, I don't know.
Thanks for watching the DNA Files.
This has been gel electrophoresis.
Tune in next week for a great episode on DNA cloning.